want to welcome everybody to the Longevity Summit. I am so glad to be here, and I am really thankful to the book for allowing us to have this great conference and for sharing their event space with us. Really appreciate it. I also want to thank all of our volunteers. So we have some graduate students and postdocs and other people from Longevity SF that are here volunteering. So thank you to them as well. And I think this is going to be a really great couple of days. I'm a little bit starstruck. Some of the really awesome speakers that are on the forefront of longevity. And I can tell you a little bit briefly about my organization. So I'm Kristen Glorioso. I'm a physician scientist, and I'm executive director of Longevity SF. We're a brand new nonprofit since last summer, and we're really focused on community building. So community is key to living a healthy life and also to solving scientific problems. And so we're connecting people in the Bay Area, and we also have a chapter in New York City, and we're looking to start chapters in other cities as well. And we've had 10 events. These are some pictures from our events that we've had so far. So we'd love to have everybody join and participate in Longevity SF as well. So thanks a lot, and I am honored to introduce Eric Verdin, who's going to be our first speaker, and he's the president and CEO of the Buck Institute here. Welcome.
Thank you, Kristen, and good morning, everyone. Who hasn't been to the Buck before? Well, that is really cool. So just a word welcome. I think if you've never been here, we've been in business since 1999. We're a not-for-profit research institute, so I'll tell you a couple of words about us, and then I'll sort of dive into the science. So the building here, as you see it, is a partially constructed building. We have 3 buildings. There are 2 more to build, so if you know someone who is interested, who has a hole burning in his pocket and a lot of resources, come and help us to build 2 more buildings. That's really the full vision of I.M. Pei, who is the architect who designed this building. If you're not familiar with I.M. Pei, he is the architect who designed the pyramids at Le Louvre and the East Wing of the National Gallery, and he lived to 102. And we told him that this is what happens when you work associated with the Buck. It sort of rubs on. What I'm particularly proud of is our workforce. We have 300 employees who are working here, all focused on the biology of aging. And we essentially cover the whole gamut, starting from the most basic aspects of understanding biology of aging in C. elegans, all the way to fruit fly, mice, and now humans. And so there's a large effort really focused on growing the mission of the Institute to address the biology of aging in humans, and including the building of a clinical center. So this is part of the vision of what we have that we will do in the future. And I hope you will meet many of them throughout the day and the couple of days. So we have a really exciting program. I'm grateful for Kristen and her organization to have asked us to host you. I think it's really part of our effort. This is a brand new auditorium, by the way. New screen, new sound system. I hope everyone hears me well. Okay, this was not always the case. And so we're quite excited to start this with this group. And nice to see also so many friends that I've known over the years. So what I'm going to do in the second part of this presentation is really go over some new biology that's emerging in my lab, and it's dealing with epigenetic clocks, as the title was highlighted. So I suspect we have somewhat of a mixed audience, so I might go deep at some points. Some other points I'll be a little more superficial in terms of the understanding, but please bear with me. I'll try to get everybody to be able to follow the whole story. So we live at really an extraordinary time in terms of our understanding of aging. In the last 20 years, we've identified a whole series of what I call dials. or flips, areas within the cell and within an organism that we can target either biologically using the tools of molecular biology or even using small molecules. And these buttons allow us to essentially regulate the rate of aging. And that's something that you would have told us this 30 years ago. Most people would have said it just cannot be done. But it is not only done, it's done regularly, and it's actually fairly easy to do. When the initial genes were identified regulating aging in C. elegans, people thought, well, this is the exception. Today, we have more than 700 genes out of the whole C. elegans genome that control aging. So this really portends to an incredibly bright future in terms of what we're going to be able to do in the future. And we really are excited to be part of this whole thing. So one example that I'd like to share of one of these buttons is TOR, target of rapamycin. So this is a cellular protein that is present in every tissue, in every organism that we have studied. It's targeted by rapamycin, the molecule that you see in the middle. Amazingly, and when you actually apply this to, do I have a pointer on this? I don't think so. When you apply this to a mouse colony, you can see the black mice are dying with a certain frequency. And the mice on rapamycin actually show a 7% increase in median lifespan and up to 20% in maximum lifespan. You can give larger amounts of rapamycin and you can see an even larger effect. And these mice actually age well. It's not that they're living longer affected by all kinds of disease. They actually show a compression of morbidity as well. Now, the amazing thing about rapamycin is that it's not only a drug that we can use in the laboratory, it's also an approved drug in the clinic. And the patients who are taking this take it at high dose as an immunosuppressant, and the field is actually working on the idea of giving this to humans at a lower dose. And I know a number of people in the field are already taking it. Everyone is taking their chances. So this is a drug that it's probably our strongest candidate. And I believe we have Matt Kaberlein here. I don't know if he's going to talk about this today. I feel a little bit sort of intimidated talking about rapamycin when he's in the audience, but I don't know. We'll see. I suspect he will say something about it. Yeah, I saw him. All okay? All right, so I mentioned there are, today over 100 interventions that can extend the health span or the lifespan of a mouse. And so, as I mentioned, a number are in the clinic already, but most of them have not been pursued. And so this is the task ahead. And I've argued for a bit of a conservative stance in this respect because once you move into humans, the barrier is much higher. As a physician, scientist, the first thing that I was told is first do no harm, and that's always going to be the thing that many of the people in the field are going to have to go by. So we can take chance on yourselves, but when you start clinical trials, we will have to be sure that first do no harm. So the barrier to translating all of these discoveries into humans is not going to be easy, but it certainly is a worthwhile effort. So some of the things that are being pursued right now, acarbose, metformin, rapamycin, senescent cell clearance actually are in clinical trials, FGF21, and so on. So the list goes on. Really exciting time in the future. And we want to be one of the major places driving this revolution in medicine. So this is all very nice, but one of the, so we have a plethora of potential drug targets and drugs. One of the biggest problems in the field is how do we conduct clinical trials targeting aging? And one of the approaches that most people have taken is to actually target specific diseases associated with aging. So you have the biomarkers of that specific disease and you target it. That's one way. The other way that a number of people are potentially exploring is the idea that we will target aging by itself. And the question then is, how do you actually measure that your drug is working without doing a 30-year or 40-year experiment, which everyone realizes will not happen? So hence the whole field of biomarkers of aging, trying to identify surrogate markers that allow you in a short period of time to determine whether a drug will have an effect or not. And I've listed on this slide a number of of properties that many of us in the field are looking for. So I should say also, we're not quite ready to start these trials. The TAME trial for metformin is probably going to be the first one that will target aging as a global disease, right, rather than that a series of individual problems. But independently, I think it's important for us to really pursue biomarkers of aging. So these biomarkers should be a reflection of a ubiquitous aging-related process. They should be universal. Hopefully, they should work across different tissues. They should be based on cellular, subcellular level process, be predictive of health span or lifespan, vary in response to interventions, and work in men, women, and population of different ethnic backgrounds. And we are far from having anything that would remotely look like this, I should say. We have, and this is what our effort has been. So there are a whole plethora in the literature. There's not a week that goes by without another paper describing a new biomarker of aging. I picked up this slide from a review in a magazine showing that when you look for two different variables, like accuracy and utility, there's one that really clearly stands above a family of biomarkers, the so-called DNA methylation clocks that were pioneered by Steve Horvath and his colleagues. And so this is what I'm going to be talking about. If you're not familiar with those, just a few words of introduction, how they are generated. So you start from a patient or a person or a group of people who have a certain chronological age and some well-defined clinical characteristics, and you regress this chronological age onto DNA methylation at CpG in the genome. So maybe this sounds like a mouthful. We have a genome, which is the DNA. On top of this is the primary code of information. This is your genetic background. On top of this, there's something called epigenetics, which actually in some way regulates the expression of different genes. And one of these epigenetic marks are these CpG DNA methylation sites. And so imagine there are millions of sites of Cs, of Gs in the genome, which actually can be methylated, and they act as a reversible switch. And whether the switch is on or off will determine whether a gene, in a very simplified manner, whether the gene is turned on or off. And so what's been observed is that these CpG sites, their frequency at different sites changes as a function of aging. And so using this type of approach, once you have constructed this, you generate an algorithm that allows you to go back and to essentially measure the level of methylation of the DNA and to actually predict the chronological age. So there's a training set using machine learning and regression type of analysis that's listed here, the lasso, elastic net. And so this becomes a tool. You can then take someone's DNA and determine where they are in their age. Now, what's more interesting is that this does not generate 100% correlation. There's space above and below the line, which means some people actually look in terms of their DNA methylation like they are younger than their chronological age, and we define this as the biological age. Some others look older, and so hence the ability to use these markers as predictive. Now, there have been a lot of these clocks generated. As I mentioned, the first one was Steve Horvath and colleagues. I've listed several of them here. Hannum's clock, PhenoAge by Morgan Levine. I believe she is here, or she's going to be here. Huh? Where is she? Okay. GrimAge and so on. And these clocks have been, are really interesting. Many of them have actually a very strong predictive value in terms of health outcomes or even mortality. One of the things that has been somewhat puzzling and frustrating has been the fact that when you look at the CpG sites that are actually regulated as a function of aging, it's very hard to discern biology. And so you can look at the genes, and it's very hard to decide what is this clock actually really measuring, and how is this related to everything we know about the biology of aging. And that's one aspect that has always bothered me, that really there has to be some relationship to biology. Number two, is, as an immunologist, quite often these clocks are conducted using whole blood. Take the whole blood from someone, you measure the clock. Now, the problem is the blood itself is comprised of more than 50 different cell types. And we know, and so many of them are listed here, and you've heard about many of them. Erythrocytes, these are the red blood cells, monocytes, macrophage, eosinophils, basophils, all the T cells, and so on, B cells. One of the things that we know also is that during the aging process, the relative abundance of these populations changes. And I'll show you actually in a minute what this looks like. And so I've always wondered, is the clock really measuring intrinsic aging it's certainly not working on a homogeneous population. Could it be that many of these cells actually have different ages independently of each other in terms of methylation, and that what we're measuring is actually a shift in populations versus and that would be one very good reason why we don't see any biology. We're looking at something that's completely unrelated to aging. That is related to aging, but indirectly, by the fact that these populations change. So one thing we try to do is to revisit this by really looking at the relative abundance of many of these cells in peripheral blood as a function of aging. And so to do this, we actually, one of the trigger of doing this was the purchase of a new machine here called a Cytec Aurora. This is what's called a spectral flow cytometer. This is a machine that allows you to do flow cytometry with 40 colors. Just before this, the machine before actually did 6 to 10. So imagine the complexity in terms of the data sets when you start looking at 40 colors. And so this allows us in a deeper immunophenotyping than conventional cell cytometry. So we started with a cohort made out of people between 20 and 82 years old. As you can see, 22 males, 22 females in the sero-negative group, and 23 in the seropositive. So why did we focus so much on CMV? CMV is a viral infection that affects many of us. Actually, by the time you're 80, most people have been infected, and it has a profound effect on the immune system. So we wanted to be sure that we were not just looking at a CMV effect, so we looked at both. And so here's some of the data we see. When you get your 40 colors, imagine this is a sort of a matrix of 40 by 40. You can do what we call a dimensionality reduction analysis using UMAP in this case, and this allows you to identify a series of blobs. Like you see the B cells in orange, the NK cells in green, monocytes in bright green, and so on. And we can then basically, using this type of analysis, to quantify the relative abundance of all these populations as a function of aging. And this is what it shows you here. We see the NK cells slowly increasing during aging, monocytes slowly increasing, CD8 T cells decreasing, gamma delta T cells, another subset. But as an immunologist, I wanted to focus on one of the most well-defined populations, which is the CD8 T cells. Just a word of why we focus on this. These cells are part of the adaptive immune response, and they are highly dependent on the thymus, shown here on the left. So this is where these cells differentiate. So at the end of the whole very long, complex process, you end up with a population of so-called naive T cells who migrate into the periphery. And in the periphery, they themselves undergo a number of differentiation steps as a function. So these cells carry what's called a T cell receptor, which is specific for bacteria, viruses, pathogens, and so on. And as they are naive, you have a large pool with very low frequency of each individual cells, but a high diversity, so that these cells are ready to tackle any pathogen that will come their way. And when they get into the periphery, if the T cell meets its antigen, or the thing that it's supposed to target, it will start proliferating and it will become from a naive and effector T cell, and eventually will become a memory T cell. This is the basis for vaccination. So when you are getting vaccinated, you're presented with an antibody, with a viral protein, which will trigger your naive T cells, and they will generate these memory T cells, which then stand ready to be reactivated much faster when the antigen is presented again. So that's the whole idea. Now, there's a hitch in this whole system, is that the thymus is one of the first aging organs in the body. By the time you're 50, most of us have lost our thymus, which means that your whole immune system has no more regenerative capacity and relies only on a pool of naive T cells, which slowly proliferate in the background to maintain the pool. And obviously, no biological system is perfect, so over time, as I will show you, and actually this is the next slide. So what we did is basically to look at each of these populations as a function of aging. And this is where we saw some really interesting differences. Look at the naive T cells, how they decrease. They decrease throughout life because thymic output decreases throughout life. But by the time you're 60, actually, you can see that your number of naive T cells is really very strongly decreased. Now, this has enormous implications. For example, think about COVID epidemic. We know that if you are 70 years old, only about 30% of people will respond to any vaccines. 70% don't respond, which is really, kind of scary if you think about it. And so this is a reflection of this decrease in naive T cells. In contrast, you can see the more differentiated cells, essential memory and the effector memory, all increase. So you have a shift from one population to the next. So the question we asked is very simple. So this is probably some of the biggest differences that you see in terms of aging in the immune system. The question that we ask is, what is the age of these individual populations? Are they all the same? Initially, I guess most people were assuming if you were to analyze them, they're all about the same epigenetic age. And so we did the experiment. We worked on 7 donors, aged 30 to 77. We sorted, so flow cytometry not only allows you to measure the frequency of different cells, we can actually sort them, separate them, and purify them to homogeneity. So we purified these naive, central memory, effector memory, and AMR cells. And then in collaboration actually with Steve Horvath and Bobby at the Clock Foundation, we did a full DNA methylation readout. And this is what we saw. And I must say, the first time I saw it, I thought, there's got to be a mistake. It cannot be this, cannot be possible. Because what the data says is that this is the average of all the donors that we looked at, looking at from naive all the way to the terminally differentiated. And you can see between the naive, who are actually typically looking about 15 years younger than the age of the donor, to the terminally differentiated, who are looking actually close to 5. So we had 15 to 20 years difference in epigenetic age in the same individual on average. And so that really spoke to the fact that there's something else going on when we do this epigenetic clock. And I should say also, I want to make sure you understand, I'm not here saying that the epigenetic clocks are wrong. They're not. Actually, they're very good at what they're doing. What we're trying to do is to increase the understanding of what they really truly represent. And I've talked to Steve on a number of occasions about this data, and he's made the case that there have been effort. We're not the first time to come up with the idea that this is a confounding variable, but and there have been different attempts actually to try to extract or deconvolute that data, the variation in subsets. However, I think this is the first time that we've seen this type of data, and really suggested that since we know that naive T cells decrease during aging and the other ones increase, given their different relative age, it's easy to understand how the clock could be actually shifting simply based on the population change. So this is what this obviously triggered a whole series of questions. One being that could we actually construct a new clock that would extract this differentiation effect and only look at intrinsic aging? And we've called those sort of intrinsic and extrinsic aging. And this is what I want to show you in the next few slides. So let me show you also one more data. If we look at donor age, and now we look at the naive T cells, you can see that all the orange dots line up about lower than the diagonal, meaning that all of the naive T cells in every donor are acting 10 to 15 years younger than the other ones, and contrast for the effector memory T cells. So this really speaks to the fact that there is intrinsic aging in the naive T cells, and from the naive themselves. So if you take a purified population, just naive, so there's no shift in population, we can still see a signature, an epigenetic signature of aging. So that told us that there are the clock is actually an admixture of two different processes, one which is differentiation, and the other one which is intrinsic aging. And so we decided to build a new clock to try to extract, to separate these two variables. And this is work of so I'll skip to this. This is work of a talented student in the lab, Alan Tomusiak, who's really been amazing at doing this. So he started, I won't go through all of the details. The only thing I want you about how we generated the new clock, I'm happy to talk about it with the aficionados in the room, but the important thing to know is, look at the upper right corner, we've removed what we call CD8 positive T cell differentiation dependent CPG. So we try to remove the marks that are linked to differentiation. And happy to discuss how we did this as well. And so this led to a new clock, a new algorithm that we can then apply to DNA methylation and try to say, okay, how does this behave? And so this is how the new clock works. First, if you look at the age that we know from the donor and its predicted age, you can see it really is it's working like most other clocks have been shown to work. So you can predict chronological age and biological age relatively precisely. If you look at the correlation coefficient, it's pretty good. Now what's interesting is when now we apply this new clock on the same samples that I showed you earlier. So here's individual donors that I've showed you earlier. Again, between naive, central, effector, and TEMRA, you can see always this line going up in most, actually in all, but sometimes with jumps. Now, when we have removed this differentiation clock, you can see that the new clock now is behaving flat. And that really was the demonstration that this new clock has extracted something from the old clock, and it's, one thing that we're working also is, could we make another clock that would be only looking at differentiation? And that work is in progress, and not been as easy as we wanted it to be. Now, what's interesting, so I showed you this new clock is insensitive to CD8 T cell differentiation. I showed you this on our data set. This is on our own data set, where we can do this on other data sets that have been published. And again, what we did here is to compare Hannum's clock, Horvath clock, Levine's clock, and our new clock, and make a delta between the predicted age of the effector memory T cell and the predicted age of the naive T cells. And if there's no more effect of differentiation, we get 0. This is us. You can see all of the other clocks are biased towards measuring this. Now again, this doesn't mean that these other clocks are not working. This is not what we're trying, because I've been told, you're attacking the old clock. I said, I'm not attacking the old clock. I think they're perfectly fine at what they're doing. They're probably better than what our clock is at measuring aging. But they are highly sensitive to T cell differentiation. The same thing, what was probably more surprising is we can take our clock and apply it to an existing data set from CD4 T cells, separated from the CD8 T cells, and it still works the same way. You can see our clock does not respond to differentiation status of the CD4 T cells. The other clocks do. Now, here's why I think this work is important. You probably have seen literature, so those of you who are in the field, claiming COVID-19 is associated with accelerated aging. Now, when you start thinking about what this means, COVID-19 is a massive infection leading to chronic immune activation and leading to a shift in these populations. So what I think is being measured, at least our hypothesis, was what It just never made any sense to me how someone could age 5 to 10 years within a 3-week infection. It just does, biologically, it doesn't make any sense. And now I think what our data is saying is that this is where the existing clock, are having problems, is that they do the same thing in HIV infection, they do the same thing in a whole series of conditions where there's chronic immune activation, rheumatoid arthritis, and actually if you look at all of the factors that accelerate the clock the most efficient way, they are all linked to a chronic immune activation. And I want to show you our new clock, when we conduct it on existing data sets, actually does not change in terms of COVID-19. Other clocks are showing different effects. Now, another thing that is important to consider, I don't have the data because we've not been able to get access to the DNA, but many of you are probably familiar with the CHIM trial. The CHIM trial was published with a lot of noise, first reversal of human aging, 2 years in patients who were given growth hormone, DHEA, and metformin. Now, I want to put a seed of doubt in your mind about what this data is really saying, because growth hormone's major effect is to induce the proliferation of naive T cells. And what do naive T cells do? They have a much younger age. So my prediction is that there was no age reversal in that trial. They simply induced naive T cell proliferation, and so that led to this paradoxical effect. And I think we should be really careful about the claims that we make as a field about, especially when we talk about things as big as reversal and aging in humans. My prediction is that data set does not prove, at least, we would like to get access to the DNA to measure it. We have not been able to so far, so if you know some of the principles and you can help me convince them, I would love to measure this. Now, here's where the clock actually gets also really interesting. We have used it on a number, trying to say now, maybe by separating these two clocks, I know you're nervous about the time, but I'm almost finished. By separating these two clocks, we decided, and this is really where most of the effort we're interested in, is really trying to look at the biology. So we've looked at senescence, for example, and this is a data set that's been published. Actually, I'm missing the reference here, but I'll be happy to give it to you, where we can actually look at early passage, near senescence, and senescence, and you can see a really nice increase in aging using our clock. And I have the data, but I won't have the time to show you. Our clock is better at doing this in senescent cells than most actually existing clocks. Remarkably also, if you take reprogramming using the Yamanaka factor, this is one way in which we know we can actually bring cells from a differentiated state all the way to a stem cell. You can see how our clock actually goes back down to 0 as a function of this treatment. So really sort of validating that it's measuring something quite interesting in terms of aging. Now, this is one of the things that happens with our new clock, and I'll go very quickly through this, is that a whole series of indications that were reported to show clock acceleration actually do not accelerate using our new clock. And many of them actually are autoimmune or inflammatory disease, celiac disease, multiple sclerosis, rheumatoid arthritis, HIV, COVID, and so on. Interestingly, our clock actually tracks very strongly with cancer. And we don't really understand why, but this really has interesting implications with Parkinson's and dementia. So I think because I've already been through a little bit over time, I just want to really quickly wrap up and just tell you what I've just told you. CD8 T-cell differentiation state plays a major role in epigenetic clock age. So we've created a new clock, which is resilient to change in T-cell differentiation, and this clock actually exhibits some interesting properties. We're really now trying to investigate the connection between the epigenetic age and measures of health span, transcription, and epigenetic changes. Again, a word of caution about interpreting results based on the other clocks, which are, again, a mixture of two different measurements. And so finally, most exciting for us is really using this new tool. We've been able to identify in our subset of genes that are that are forming the basis of this clock. And when you look at their biology, it's much easier to understand how they are related to aging. At least that's what the preliminary result do. So we were planning actually to use this clock also as a tool for screening in vitro for small molecules that revert this clock. So this has opened up a whole series of interesting collaboration and processes in the lab. I want to finish by recognizing the work of the people in the lab who've done this. Alan Tomusiak, the graduate student, I've mentioned him. Herb Kassler, who's directing our flow core here at the Buck Institute, who's been a long-term partner in working on immunology. Hiroyuki Matsui, who generated the subset analysis, and Marlene Cervantes, who's worked with all of them closely. Thank you.
Of your big picture, first figure from your new clock, in which you show the correlation with chronological age, and you're referring to the scatter as biological age. In fact, that scatter, from first principles, is human variation and error, and is only a hypothesis about biological age, just as in the case of the Greg Fahey human growth hormone, I have a monthly Zoom call with Horvath and Bobby, in which I told Steve that given what we know about growth hormone accelerating aging, that if I had the result that use of growth hormone lowered the age clock, I would conclude that my age clock didn't measure aging. So what would you say about the obesity and smoking aspect, and where do we stand with really thinking about biological age?
So for us, I think we've looked at obesity and smoking, and they don't move with the clock. Now, people have been using the argument to say that's accelerated aging, but I think we have to be really careful when we talk about accelerated aging. Having a heart attack is accelerated aging because you die. So, to me, there's something else. When I speak about accelerated aging, I really speak about not living shortly. I think about sort of having some of the intrinsic pathway that regulate your rate of aging change.
Smoking does that?
Huh?
You don't think that smoking does that?
Smoking increases lung cancer. Smoking has a lot of really toxic effect. Alcohol also makes you live shorter. Is that accelerated aging? So I think, a lot of people have used the fact that the clocks is accelerated to say, oh, that's accelerated aging because they're living shorter. But not everybody who lives shorter, in some way shows accelerated aging, but I'm speaking from a different perspective. These pathways that are regulating. Does that make sense? And so, I should say also, we're still early. And now, one other thing that's important to realize is that the existing clock are important tools. And we're not trying to replace the existing clock with this clock. We just want to understand the biology that understands these variations. Because the truth of the matter is, during aging, there is a shift. The earlier you have thymic involution, the earlier you see the shift to less naive T cells and so on. So they're measuring something that is clearly related to aging. But we call it extrinsic aging versus intrinsic aging, which is the type of curve that we see only in naive T cells.
Thank you so much.